for the very gracious introduction, and I thank the organizers for giving me this opportunity to tell you about a story that we published last year. And I want to tell you this story because this highlights the unique challenges of developing a vaccine against dengue. Okay. So since most of us in the Western world, we know very little about this virus, I figured I'll give you a little introduction to this virus first. This virus, dengue virus, causes a spectrum of illnesses in humans ranging from dengue fever to dengue hemorrhagic fever and dengue shock syndrome. Dengue fever is also known as breakbone fever because of very high fever and the pain associated with headache. These joint and muscle pains are so severe that people just, in the endemic countries, they call it the breakbone fever. And another feature of dengue fever is rash. The next severe form of the infection is called dengue hemorrhagic fever. In terms of hemorrhagic manifestations, most patients have eye, nose, and gum bleeding. And this hemorrhagic fever, this DHF, is really a misnomer because the hallmark of dengue hemorrhagic fever is this phenomenon called plasma leakage syndrome, which is leakage of fluid from blood vessels into tissues. And if this plasma leakage syndrome becomes severe enough, then the patients undergo shock. And at this case, then yes, you do have massive intestinal hemorrhage and dengue shock syndrome or DSS, it can be fatal. So in terms of, again, this is to emphasize the point that the hemorrhagic manifestations, these are the more typical ones, rash, petechia, and such as eye bleeding. And the hallmark is really this plasma leakage syndrome. If it becomes severe enough, the patient undergoes, the circulatory system of the patient collapses and they go shock. So what kind of virus is it? Well, there are 4 different serotypes of these viruses, and these are transmitted to humans by these mosquitoes. Shown here is a picture of this mosquito called Aedes aegypti. And in the Asian world, we also call them as the tiger mosquitoes because of their beautiful stripe patterns. Shown here is the geographic distribution of these dengue-transmitting mosquitoes in blue. And in the red represent countries that have both these dengue-transmitting mosquitoes as well as dengue epidemic activity. So the point of this figure is virtually almost all subtropical and tropical regions of the world are affected by this virus. In terms of dengue as a quantitative problem, we have over an estimated 3 billion people in the world who are at risk for infection with this virus. Out of those 3 billion people, an estimated 50 million cases of dengue fever occur each year. Out of those 50 million, about 250 to 500,000 go on to develop the severe dengue disease, DHF, DSS. And out of these people, about 24,000 die each year. Despite this global morbidity and mortality associated with this virus infection, I have to point out that, especially for this audience, at present, we don't have dengue-specific vaccines or antivirals. So what kind of virus is this? It belongs to the genus Flavivirus. The other medically relevant viruses of this genus include the West Nile, the Japanese encephalitis, and yellow fever. And this entire genus belongs to this family, Flaviviridae. And of course, the most famous Flaviviridae member is the hepatitis C virus. These are small envelope viruses, about 40 to 60 nanometer in size, and their genome is less than 11 kb, and they're single-stranded positive polarity. What that means here is, here I'm showing you the schematic of this viral genomic structure. My little squigglies are the 5' and the 3' untranslated regions. So this viral genome is translated, is also the messes, and is translated as one big polyprotein. And both the viral and the host factor proteases are involved in cleaving this one big polyprotein into several proteins. So in terms of the structures, the C, M, E are the 3 structural proteins known as the capsid, membrane, and the envelope. And NS1 and 5 represents the non-structural proteins. And so these proteins often have multiple functions. For example, NS3 acts both as a protease and helicase, and NS5, the biggest protein, is the RNA-dependent polymerase. So I was attracted to the dengue field, and dengue is famous in the virus world because of its very interesting epidemiology. And what that is that several epidemiologic studies have shown that secondary infection with a different serotype is the single greatest risk factor for coming down with the severe form of the disease. So basically, again, to emphasize what people have shown is that most severe cases of this DHF-DSS, they're observed in patients with secondary infection. And another interesting epidemiological data have shown that in infants, all infants with severe dengue disease, people have found that they're born to dengue immune mothers. And people were able to correlate the levels, the low levels of dengue-specific maternal antibody to these infants with severe disease. So based on these two pieces of epidemiologic data, people have come up with one of the hypotheses. There are several hypotheses that have been proposed to explain how people get the severe disease. But one of the major hypotheses in the field is called antibody-dependent enhancement, or ADE for short. And this was proposed almost 50 years ago. So what is ADE? So ADE is essentially when you have subneutralizing levels of antibody, and so the red here is represents the virus and the antibody. And remember, this could be due to previous infection or from dengue immune mother in the case of infants. So you have this virus antibody complexes. They get into cells through these Fc receptors. So these are cells that are normally not permissive to dengue infection, but now through this Fc receptor interaction, you have increased viral load. And we don't quite really understand how somehow this increased viral load, either through T cell activation or through directly because of this increased viral load, we have this phenomenon known as cytokine storm, and that's basically your loose term people like to use because dengue patients have high levels of both pro and anti-inflammatory cytokines. And somehow this cytokine storm is believed to trigger vascular leakage, the plasma leakage syndrome, shock, and ultimately death. So that's the ADE hypothesis. So when we entered the field, we decided, okay, let's assume this is a major hypothesis in the field. Let's assume this is real. So what would that mean in terms of dengue infection? And so what it would mean is that if you would be infected during primary infection, you develop a nice antibody response, and you generally get a mild or subclinical disease. Now, in the case of if you get reinfected with the same serotype, remember there are 4 different known serotypes of these viruses, the antibody response that you develop during the primary infection will be neutralizing and you'll be protected. Now, the interesting scenarios happen when you have secondary infection with the heterologous serotype. And so my blue is this purple is the different serotype. So not all of the antibodies that were generated against the primary infection virus will be able to bind, and you have this self-neutralizing condition, and that will lead you through this ADE phenomenon. You'll have the severe disease. And now in the case of infants, and now for in terms of the audience in the field, you can imagine certain vaccines. And so in the case of these maternal antibodies and certain vaccine-induced antibody response, they may also be subneutralizing and cause an ADE phenomena, and then you'll have this severe disease. So with that as background, speaking of team, this is the scientific team. 2 highly talented people in my lab, Raphael Zellweger, a postdoc who's actually here in the audience, and Tyler Prestu, and again, another very highly talented student in the lab. They decided to tackle this ADE hypothesis using mouse model in my lab. So again, just the rationale was that there's this evidence for ADE based on this epidemiologic data and numerous in vitro studies, but in vivo, despite 50 years of research, the ADE had never been demonstrated in vivo. So they set out to see if they could develop a mouse model and show that is that really sufficient to cause severe disease. And if we have a mouse model, of course, that's a home run. We can now really begin to understand how ADE really works. In terms of an experimental idea, it's actually a very simple idea. So we take this mouse, we call it the AG129 mouse. It lacks both type 1 and type 2 interferon receptor. And the reason we have to use this mouse is because wild-type mice are highly resistant to dengue infection. Then we use this particular viral strain. We call this S221. It's a mouse-adapted dengue 2 strain. And at this particular dose, through this IV route of infection, these mice develop ruffled fur, signs of sickness around day 4 and 5 post-infection. Around day 10 and 12, they develop paralysis, and as per our animal protocol, we're scoring this paralysis as death. And now I want to remind you that paralysis is not a relevant phenotype in humans, and so that's going to be important later on. And so this is what happens. So the big question is, what happens when we give them dengue-specific antibody? How does this normal course of infection changes to this virus infection? In terms of the actual experiment, we took these mice, we injected them 15 micrograms of dengue-specific monoclonal antibody. A day later, we infected the mice with our virus, and a day later, we injected the same amount of antibody and then looked at survival. Shown here are results of experiments using 3 different monoclonal antibodies. And so here we have this with 2 different isotypes, so IgG2A, and this one is IgG1. And they're also with different specificity. So this one is cross-reactive, recognizes all 4 serotypes. This one is also cross-reactive, recognizes all 4 serotypes. Plus, it's actually pan-flavivirus reactive, so it recognizes Japanese encephalitis, yellow fever, and all the other flavies. And this one is actually serotype specific, okay? So this is this virus that we're challenging, remember, is also dengue too. And what we see here is that, as expected, the mice injected with the isotype control antibody, most of them develop paralysis around day 10 to 14 post-infection. So this is our isotype control. And now what was really exciting for Rafi and Tyler was that when mice were injected with this antibody at this concentration, they developed this severe lethal disease within the first, most of them died within the first 4 days of infection. And what's important now is that none of these mice exhibited signs of paralysis. So this is really a very important development for the field in terms of having a mouse model on not showing a paralytic phenotype. So this shows that antibodies can enhance the severity of disease. So I'm sure the immunologists in the crowd were thinking, well, she's injecting this one concentration of antibody. What happens when you give them different doses? So we can think of the relationship between antibody concentration and disease in terms of this overall antibody binding, which, as we know, is, I mean, the experts are here, dependent on both concentration and affinity. So you can imagine if you have extremely low levels of antibodies because of very low concentration or affinity, it's almost like having no antibodies. So we call this a negligible effect. Now, if you have the same antibody, it's low concentration or low enough affinity so that you have a subneutralizing condition, then you would imagine you get the ADE phenomena. And then now the same antibody, if you have sufficient enough concentration and affinity, now you should have protection. Again, I am a mouse lab. Like my lab is, the whole world revolves around mice, so we like to think in terms of mice. And here, this is the mouse infected in the absence of antibody, and this is the normal course of infection. Now, if you give them this extremely low, negligible amounts of antibody, it shouldn't be any different from this first scenario. Now, if you give them the subneutralizing concentration of antibody, you see this is enhanced lethal disease at very early time post-infection. And if our hypothesis is correct, the same antibody, if you give them high enough, the mouse should be protected. So do we see that in our mouse model? And of course we do, otherwise I wouldn't be showing you this data. The experimental setup is the same, which is we take the mice and inject them with different concentrations of antibody, again, infect them, and then second dose. So instead of showing you percent survival curves, here I'm showing you the mean survival time on the y-axis. And here we have the PBS and the isotype. These are our non-ADE mouse controls. And at this 0.5 microgram of antibody, so that's like a negligible concentration. So there's really no difference between the non-ADE mice and this 0.5 microgram antibody dose. Now, when we give them 2 or 15 micrograms of antibody, we see this ADE effect, like enhanced this early lethal death phenotype. And now, if you give them this high enough dose, 500 microgram, we can actually rescue the mouse from the disease. So this is an important concept, not for the antibody field, and which is the same antibody can have both an enhancing and a protective effect. So this time is up. So remember, the key feature was most of the animal mouse models to date, they showed the paralysis wasn't a relevant phenotype. We wanted to see if these mice in our ADE model, they exhibited key aspects of the human disease. And sure enough, so we looked at various parameters that are associated with severe dengue disease in humans. And one of them is, remember, is cytokine storm. So we looked at levels of, these are the 3 major cytokines that have been implicated in dengue pathogenesis. We looked at IL-6, IL-10, and TNF. And sure enough, these mice have very high levels of all 3 cytokines, especially towards this when they're about to die at 90 hours post-infection. And most interestingly, we can inhibit TNF and rescue the mice from this lethal disease phenotype, but not when we inhibit the IL-6 or the IL-10 receptor signaling. So what this shows is this lethal disease phenotype is mediated by TNF. Another key aspects in humans with dengue disease, they have low platelet count and they have elevated hematocrit. And sure enough, in our mice, these are ADE mice as compared to the non-ADE control mice, they have low platelet count and elevated hematocrit. And of course, remember I made a big point about the hallmark being the vascular leakage, and we performed a standard vascular leakage assay in the animal model. So we looked at the levels of Evans blue in tissue. So we're just showing you one tissue here, the liver. And again, the ADE mice have higher levels of Evans blue in the tissue, meaning vascular leakage as compared to the controls. And again, remember I told you about intestinal hemorrhage being hallmark of the severe dengue disease. And sure enough, this is our non-ADE mice. We don't see intestinal hemorrhage, and now we see intestinal hemorrhage in our ADE mice. And now that we felt comfortable that, like, okay, our mouse model really has these key features of the human disease, it was now here comes the fun time. Can we begin to understand this mechanism of ADE? So one of the first questions we ask is, as predicted by the hypothesis, does ADE result in increased viral load in tissues? And so we looked at virtually every single tissue in the mouse, and I'm showing you results of only four of the tissues that's been implicated again in human dengue infection. And we see that, yes, again, this is the QRT-PCR data. And we see that compared to the non-ADE mice, the open circles, the ADE mice always have significantly higher viral load. And what's most interesting is that liver stood up right away for us, because this was the tissue where we saw the earliest difference between the non-ADE and the ADE mouse. So starting at 48 hours post-infection here, this time point, this is when we first see the difference. And then only at later time points in these other tissues, we see like 72 or 96 hours, we see this other higher viral load in the ADE mice. The next feature of these, in terms of mechanism of ADE, is the hypothesis predicts that it's an interaction between the FC receptor and the FC portion of the antibody. So we wanted to see if that was the case in vivo as well. And remember, because of liver, so in terms of the survival and death phenotype, we decided to look at our readout here. It's this viral load in the liver at 48 hours post-infection. So this is our non-ADE mice, the mice injected with the isotype control antibody. This is the ADE mice, so this is the amount of virus we see in the liver. And now, believe it or not, if you flood the mouse with 2,4-G2, this is a reagent that we use for staining, it actually blocks FC receptors in vivo. And now we see this low viral load, the non-ADE type of response. We can use the Fab of that antibody. Again, now we have no ADE. And we could actually restore ADE in these mice by coupling the Fab to the isotype control here. So again, just as predicted by the hypothesis, ADE does involve interaction between FC portion of the antibody and FC receptors in cells. So again, like now for us, like, okay, those are all the predictive things and that it works. So it looks like we really have an ADE model. We wanted to explore, it's like, okay, another big key question in the field is who really are these cell types that are supporting ADE infection of dengue? So remember, liver is our major tissue, so we wanted to answer this question by performing flow cytometric analysis. So we looked at, we isolated the liver cells and stained them for CD31, which is an endothelial cell marker, and CD45, which is a hematopoietic marker. And based on literature, again, almost 50 years of literature, we expected these dengue antigen expressing cells to be in the R2 gate, which are these macrophages, dendritic cells in the liver. But to our surprise, we saw that both at the 48-hour and the 72-hour time point, we see that in the ADE mice, the dengue antigen expressing cells fall in this R3 gate. So these are CD31 positive cells, and then in the liver, most of the CD31 positive cells are LSAC, which stands for liver sinusoidal endothelial cells. So because of time's sake, I'm going to move quickly. The caveat of this experiment is that the antigen that we use detects the structural portion of the antibody. So we wanted to be confident that we're really looking at viral infection. So here we use a different approach, immunohistochemistry, and a different antibody that really measures the viral replication. So we're NS3. And we see that by this measure again, so non-ADE, ADE, non-ADE, ADE mice, the dengue antigen expression in the green co-localizes not with CD68, the F480, but CD31 here. So again, by two different approaches, we show that these are liver sinusoidal endothelial cells. So this is completely really against the dogma, what's been reported in the literature. So in summary, what we've shown that we take a mouse, we inject them with antibody or immune serum and virus. We've shown that this mouse gets sick. One of the first things we see that we have increased viral load in a variety of tissues. And the first tissue to have increased viral load starting at 48 hours is the liver. And through our flow cytometry and immunohistochemistry data, we show that this is due to LSEC infection. We show that these mice have cytokine storm, and they have vascular leakage and gastrointestinal hemorrhage, and ultimately the mouse dies because of TNF. So in terms of dengue vaccine challenges, I think the results that I just showed you, and also this result has been confirmed by another independent group, Eva Harris's laboratory at UC Berkeley. So it's not just my laboratory. We have to really think about ADEs, the potential for ADE in terms of dengue vaccine development. And so, and the reason it's a problem is because remember these are four, right now it's, what's known is that these viruses exist as 4 serotypes. And several companies are trying to develop this balanced tetravalent formulation, and I have to say that to date they haven't been successful. Even the ones that are about to, that have already started phase 3, at best they're trivalent, but none of them are tetravalent. Okay, and then I think from the dengue perspective, academic perspective, it's really naive for us to think that in the future there will only be 4 serotypes. We know that dengue exists in the sylvotic cycle between non-human primates and the mosquitoes in the forest. And you can imagine with us humans encroaching the habitat of these non-human primates, it's just a matter of time that we'll probably have another serotype. And who knows, in 5, 10, 20 years from now, we may have serotype 5, 6, and 7. So I think in terms of vaccine development, we need to think about that issue, not just the currently existing 4 serotypes. And I think, and the reason I highlighted these in blue is because this is something unique to dengue, but I think in the black is something that's not really unique to dengue. That's a problem in almost all infectious diseases. Here is like we don't really understand the mechanisms of protection, or in the case of dengue, pathogenesis, both from the host immune side and the virus side. And of course, because this is a mosquito-borne disease, the epidemiology of this is it's still rudimentary. We don't really understand the interaction between the mosquitoes and the virus. I just told you we don't understand the interaction between the mosquitoes and the humans, so now imagine, and also the interactions between the mosquitoes and the humans. So it's quite complicated, and I think we're just beginning to understand that. But I'm not really here. I wasn't really invited to tell you all the bad things. But I wanted to tell you that I think what this calls for is a tremendous opportunity in terms of developing novel vaccines to solve the global dengue problem. And of course, it's not a surprise to you that coming from animal model perspective, I think this development of new animal models are very exciting, both in terms of developing and testing dengue vaccines. And I think we should know that we have flavivirus vaccines, such as the ones for yellow fever, and Japanese encephalitis that work. And in fact, very notable immunologist includes Valley Plendrin uses the immune response to yellow fever vaccine to understand fundamental mechanisms of the immune system. So we shouldn't be discouraged that, oh my God, this dengue is a crazy problem. And of course, NIH and various nonprofit organizations, they're beginning to recognize dengue as a big problem. And I think in the next, we're already beginning to show the results of their, recognizing this as a category A pathogen and increasing funding. And similarly with the Gates Foundation, through their support, there are numerous cohorts that have, that are really basically ready so that dengue vaccines could be tested. So let me tell you, so this is actually the most important slide. I told you the story of a very talented postdoc, Raphael Zellweger, and talented student Tyler Prestud, and they were supported by my lab works as a team, all these members in my team, and I'm sure at Crisal you know about Lauren and Stuart. They just joined you recently, and of course the funding from the NIH. Thank you for your time.